Good evening and welcome to Dreamy Academy. Tonight we begin a quiet journey into the very architecture of life itself, exploring the hidden dance of molecules that makes us who we are. As we begin, I'd love to know where you're watching from, your city, your country, perhaps even what the night sky looks like outside your window. If you enjoy these peaceful explorations of science, please take a moment to like the video and subscribe. It truly helps this little corner of quiet curiosity grow. To those already subscribed, thank you sincerely for being part of this slow and thoughtful adventure. Right now, as you listen, as your breath gently rises and falls, an astonishing universe is unfolding within you. Not a universe of distant stars or swirling galaxies, but one infinitely smaller, yet no less profound. Within every cell of your body, a symphony of unseen forces is playing out, a ballet of tiny particles and ephemeral fields. Your thoughts, your memories, the very beating of your heart, all are intricate expressions of chemistry, of molecules interacting with a precision and elegance that defies easy comprehension. We often think of ourselves as solid, tangible beings, defined by the boundaries of our skin. But if you could truly see, if your gaze could shrink beyond the visible, past the cellular, to the molecular scale, you would discover a universe of vast emptiness, punctuated by fleeting arrangements of energy. You would see that the very substance of your being is a delicate, dynamic tapestry woven from the most fundamental units of matter, constantly rearranging, constantly communicating, constantly alive. This isn't just science, it is poetry written in the language of atoms, a silent epic unfolding in every moment. To explore molecular biology is to gaze into a mirror and see not just a reflection, but the shimmering, intricate machinery that makes reflection, and indeed, all life possible. It is a journey that promises not just knowledge, but a profound and gentle wonder, a quiet awe at the intricate beauty of existence. Consider for a moment the ordinary miracle of a single drop of water resting still on a leaf. It seems simple, transparent, yet within its shimmering curve lies a universe of molecular motion, a quiet, ceaseless dance. To truly understand life, to grasp its essence, we must first learn to listen to the whispers of the very small, to descend past the familiar world of our senses and enter the realm where matter itself begins to take shape. We begin with the most fundamental question: what is everything made of? And how do these building blocks arrange themselves into the astonishing complexity we call life? Let's begin by considering the atom, the basic unit of all matter. It's a word we hear often, a concept we learn early, yet its true nature often eludes us in its simplicity and its profound strangeness. Imagine an atom not as a tiny solid sphere, but as a vast, mostly empty space. At its heart lies a dense, positively charged nucleus, a tiny sun containing protons and neutrons. Around this nucleus, electrons, those fleeting negatively charged particles, do not orbit in neat planetary paths as we might once have pictured. Instead, they exist in shimmering clouds of probability, quantum whispers of where they might be found rather than where they are. The atom, then, is less like a miniature solar system and more like a resonant bell, vibrating with potential. Its form defined by the delicate balance of electromagnetic forces. If an atom were the size of a cathedral, its nucleus would be a tiny fly buzzing at the center, and the electrons would be like motes of dust, almost infinitely small, defining the vastness of the empty space. Yet it is this vast emptiness, this delicate balance, that gives rise to all solid form, The elements that make up our world, and indeed our very bodies, are simply different types of atoms distinguished by the number of protons in their nucleus. Hydrogen, with just one proton, is the simplest and most abundant element in the universe. Carbon, with 6 protons, is the elegant architect of all life, its unique properties allowing for the vast array of complex molecules we will explore. Oxygen, with 8, is the breath of our world. These fundamental differences, dictated by the number of protons, bestow upon each element its unique identity, its particular way of interacting with the world. Now, these individual atoms, like shy dancers, rarely exist in isolation for long. They are drawn to each other by an invisible longing, a fundamental drive to achieve a more stable state, often by completing their outer shells of electrons. This mutual attraction, this silent agreement, is what we call a chemical bond. And it is in the formation of these bonds that the true magic begins to unfold. Think of it like this: electrons are shared or exchanged between atoms, forming connections that are both powerful and exquisitely specific. When electrons are shared, like 2 hands clasping in a gentle but firm grip, we have what's called a covalent bond. This is the strong, stable glue that holds most of the molecules of life together. Carbon, in particular, is a master of forming 4 such covalent bonds, allowing it to build long, intricate chains and rings, the very backbone of organic chemistry. Other atoms, like oxygen and nitrogen, also form covalent bonds, adding their own unique flavors to these molecular structures. Sometimes electrons are not shared, but rather given and taken, like a small gift exchange between atoms. This creates an ionic bond, where one atom becomes slightly positive and another slightly negative, drawing them together like tiny opposite poles of a magnet. These bonds are often found in salts, like the sodium chloride that seasons our food, but they also play crucial roles in the delicate electrical signals that course through our cells. It is these bonds, covalent and ionic, strong and subtle, that allow individual atoms to assemble into molecules. A molecule, then, is simply a stable arrangement of atoms held together by chemical bonds. Water, that seemingly simple drop on a leaf, is not just H2O, it's a small, bent molecule where 2 hydrogen atoms are covalently bonded to 1 oxygen atom. This seemingly simple architecture, however, gives water its extraordinary properties, making it the very solvent of life, the medium in which all molecular ballet takes place. As we gently drift further into this molecular world, we begin to see that these fundamental principles, the structure of atoms, the nature of chemical bonds, are not just abstract concepts. They are the invisible threads from which all biological complexity is woven. From the simplest sugar to the most intricate protein, every living thing is a breathtaking testament to the elegant rules that govern the very small. We are, in essence, walking, breathing, thinking collections of molecules, brought together by these precise and beautiful interactions. There is a deep comfort in this understanding, a quiet realization that the universe, even at its most microscopic, is built upon patterns of profound order and exquisite design. In our last gentle exploration, we began to uncover the hidden dance of atoms, those fundamental whispers of the very small, and how their elegant partnerships, through the sacred vows of chemical bonds, weave the initial threads of existence. We marveled at carbon, the versatile architect, and glimpsed the simple yet profound structure of water, H2O, a molecule whose unassuming appearance belies its extraordinary power. Now let us draw closer, for it is water, the very essence of life's fluidity, that beckons us to understand its deeper secrets. Imagine, if you will, stepping into a vast, shimmering ocean not of blue waves, but of microscopic interactions. This is the world within every cell, within every living thing, and its primary medium is water. We know its formula: 2 atoms of hydrogen, 1 of oxygen. A seemingly straightforward arrangement, yet within this simple geometry lies a secret that unlocks the very possibility of life as we know it. Think back to those covalent bonds we discussed, where electrons are shared between atoms. In water, the sharing isn't quite equal. Oxygen, with its greater pull on electrons, a property we call electronegativity, draws the shared electrons closer to itself, like a benevolent but slightly possessive parent. This creates a subtle but profound imbalance. The oxygen atom acquires a slight negative charge, while the hydrogen atoms, having their electrons pulled away, bear slight positive charges. This uneven distribution of charge transforms water into a polar molecule, a tiny magnet with distinct positive and negative poles. Can you feel it? This subtle polarity is not a flaw, but a design of exquisite genius. It means that individual water molecules, though electrically neutral overall, are constantly reaching out, forming fleeting yet vital connections with their neighbours. The slightly positive hydrogen of one water molecule is irresistibly drawn to the slightly negative oxygen of another, creating a delicate embrace known as a hydrogen bond. These aren't the strong, unyielding, covalent bonds that hold atoms together within a molecule, but rather gentle, transient attractions between molecules, like hands briefly clasping and releasing in a slow, intricate ballet. And it is this dance of hydrogen bonds, these ephemeral yet ubiquitous connections, that grants water its miraculous properties. Let's consider a few, for they are the very scaffolding upon which life is built. First, cohesion and adhesion. Water molecules, bound by their hydrogen bonds, tend to stick together. This is cohesion, the attraction of water to itself. It's why water forms drops, why it has surface tension, brise, that almost invisible skin allowing a water strider to walk across its surface, or why a full glass of water can be mounded slightly above the rim. And adhesion. That's water's ability to cling to other surfaces, especially those that are also polar. Together, cohesion and adhesion allow water to climb, defying gravity in a process called capillary action. Think of how trees draw water from their roots to their highest leaves, or how blood moves through tiny vessels. It's the silent upward march of hydrogen bonds. Then there's water's remarkable ability to moderate temperature. Have you ever wondered why coastal regions often experience milder climates than inland areas? Or why your body maintains a relatively stable temperature even when the outside world fluctuates wildly? We can thank water's high or specific heat capacity. It takes a significant amount of energy to raise the temperature of water, because much of that energy is first absorbed to break those numerous hydrogen bonds before the molecules can begin to move faster and heat up. Conversely, as water cools, those bonds reform, releasing heat slowly. This property makes water a magnificent thermal buffer, stabilizing temperatures both globally and within our own biological systems. And when water finally does absorb enough energy to evaporate, it takes a vast amount of heat with it. This is its high heat of vaporization. It's why sweating cools us down. As water evaporates from our skin, it carries away excess body heat, a beautifully efficient biological air conditioning system. Perhaps one of water's most famous anomalies is its density. Most substances become denser as they cool and solidify, but not water. As it approaches freezing, water molecules arrange themselves into a more ordered, crystalline lattice, where the hydrogen bonds hold them slightly further apart than in liquid water. This makes ice less dense than liquid water, which is why ice floats. Imagine a world where ice sank. Lakes and oceans would freeze solid from the bottom up, trapping and killing aquatic life. The floating ice acts as an insulating blanket protecting the life beneath. It's a subtle difference with monumental implications for the biosphere. Finally, and perhaps most crucially for our journey into molecular biology, water is the universal solvent. Its polarity means it can dissolve a vast array of substances, especially those that are also polar or ionic. When you dissolve salt, sodium chloride, in water, the slightly negative oxygen atoms of water cluster around the positively charged sodium ions, while the slightly positive hydrogen atoms surround the negatively charged chloride ions, pulling them apart and dispersing them. These dissolved substances, now free to move and interact, are the reactants in countless biochemical reactions. Water is the sage, the medium, the very matrix in which the drama of life unfolds. Without it, the intricate chemical symphony of our cells would simply grind to a halt. So that as we observe this humble molecule, we see not just H2O, but a silent architect, a gentle protector, and an indispensable facilitator. Is the solvent of life, the cradle for all subsequent molecular complexity. Now, with water setting the stage, let's turn our gaze to the stars of the show, the intricate players that water helps to bring together, the organic molecules. Remember our elegant architect carbon? We learned of its extraordinary ability to form 4 stable covalent bonds. This unique versatility is the bedrock of organic chemistry, the chemistry of life. Carbon atoms can link together in seemingly endless ways, forming long chains, intricate rings, and complex branched structures. It's like having a set of building blocks that can snap together in an infinite variety of patterns, each one capable of holding different decorations. These decorations are what we call functional groups. Imagine a basic carbon skeleton, a simple chain or ring of carbon atoms. This skeleton provides the structural backbone, but it's the specific clusters of atoms attached to it, these functional groups, that truly give the molecule its personality and dictate how it will behave in the watery environment of a cell. Let me introduce you to a few of these crucial molecular adornments, for they are the keys to unlocking a molecule's function. The hydroxyl group, OH. Picture an oxygen atom bonded to a hydrogen atom and then attached to a carbon skeleton. Molecules containing hydroxyl groups are called alcohols, and they tend to be polar, increasing a molecule's solubility in water. Think of sugars. They are rich in hydroxyl groups, which is why they dissolve so readily. The carboxyl group, COH. Here, a carbon atom is double bonded to one oxygen atom and single bonded to another oxygen, which in turn is bonded to a hydrogen. This group is acidic, meaning it can donate its hydrogen ion in solution. It's found in amino acids, the building blocks of proteins, and gives them their acidic character. The amino group, NH2. A nitrogen atom bonded to 2 hydrogen atoms and then to a carbon skeleton. This group is basic, meaning it can accept a hydrogen ion from solution. You'll find amino groups too in amino acids, giving them their basic character. The phosphate group, OPO32. A phosphorus atom bonded to 4 oxygen atoms. This group is highly energetic and plays a central role in energy transfer within cells. Think ATP, the energy currency of life. It also forms the backbone of DNA and RNA. The sulfhydryl group, S. A sulfur atom bonded to a hydrogen atom. This group is important in stabilizing protein structure, forming what are called disulfide bridges that act like molecular staples, holding parts of a protein together. The carbonyl group, CO. A carbon atom double bonded to an oxygen atom. If this group is at the end of a carbon chain, it's an aldehyde. If it's within the chain, it's a ketone. Sugars often contain carbonyl groups. These functional groups are not mere accessories. They are the active sites, the reactive centers, the very language of molecular interaction. They determine whether a molecule is acidic or basic, polar or nonpolar, how it will interact with water, and which other molecules it will recognize and bind to. They are the molecular hands that clasp, the eyes that see, the voices that whisper instructions within the cellular symphony. As we move from the foundational atoms and simple molecules like water, we begin to see how these building blocks don't just exist in isolation. They come together, guided by the principles of bonding and the specific properties conferred by functional groups to form larger, more complex structures. We call these grander constructions macromolecules, and they are the true giants of the biological world. Imagine constructing a magnificent cathedral. You start with individual bricks, atoms, then combine them with mortar to form walls, simple molecules. But to build the soaring arches and intricate spires, you need to assemble these smaller units into much larger, more complex components. This is precisely what happens in biology. Most macromolecules are polymers, long chains built from repeating smaller units called monomers. Think of a string of pearls. Each pearl is a monomer, and the entire necklace is a polymer. Life builds its incredible diversity and complexity by linking these molecular pearls together in specific sequences and arrangements. How do these polymers form? Through a beautiful process called dehydration synthesis. It's a bit like two hands joining, and as they do, a tiny molecule of water is released. When two monomers join, a hydroxyl group is removed from one and a hydrogen atom from the other, forming a water molecule and creating a new covalent bond between the monomers. It's a precise, energy-driven construction. And how are these polymers broken down when their work is done, or when the cell needs their constituent parts for something new? Through the reverse process, hydrolysis, which literally means water breaking. Here, a molecule of water is added, breaking the covalent bond between monomers and separating them. It's a dynamic, constant cycle of building up and breaking down, reflecting the ceaseless activity of life itself. These principles of monomer-polymer assembly are universal across the four major classes of biological macromolecules: carbohydrates, lipids, proteins, and nucleic acids, each playing a distinct and vital role. For instance, consider carbohydrates. The sugars and starches that are primary energy sources and structural components. Their monomers are simple sugars, or monosaccharides, like glucose. Link many glucose molecules together through dehydration synthesis, and you form a complex polysaccharide, like starch, for energy storage in plants, or glycogen, for energy storage in animals, or even cellulose, for structural support in plant cell walls. Each chain, each arrangement dictates a different function, a different role in the grand design. As we gently close this chapter, I invite you to pause and reflect on the quiet elegance of these processes. From the subtle polarity of a water molecule to the intricate linking of monomers into vast polymers, we are witnessing the fundamental architecture of life being laid bare. It is a testament to order, to interaction, and to a breathtaking ingenuity that unfolds ceaselessly within and around us. The stage is set, the solvent is ready, and the building blocks are poised. In our next exploration, we will delve deeper into the specific stories of these magnificent macromolecules, discovering the profound beauty and purpose woven into each thread of existence. Having glimpsed the simple elegance of carbohydrates, those sweet and structural architects of life, we now turn our gaze to other equally vital players in this grand molecular ballet. Imagine, if you will, that we are still in the quietude of our dreamy academy, observing the universe unfold in miniature, marvelling at the artistry of existence. We've seen how monomers link to form polymers, a principle that echoes through the very fabric of life. Now, let us continue our journey, moving from the sugars to the elusive water-fearing molecules that define boundaries and store vast reserves of energy. As we delve deeper, we encounter a class of molecules that, unlike the water-loving carbohydrates, shy away from our universal solvent. These are the lipids, a diverse group united by their defining characteristic, their profound distaste for water, or their hydrophobicity. Think of oil separating from water. This is the essence of lipid behavior. They are the silent, shimmering barriers, the hidden storehouses of energy, and the subtle messengers that guide cellular conversations. Consider first the triglycerides. Perhaps the most familiar form of lipid, often simply called fats and oils. These are molecules of elegant simplicity, formed when a single glycerol or molecule, a small 3-carbon alcohol, extends its 3 hands to clasp 3 long hydrocarbon chains known as fatty acids. This union, like all macromolecule formation, is a dehydration synthesis reaction, releasing water as the bonds are forged. A fatty acid chain is a fascinating thing. A long, slender ribbon of carbon atoms, each adorned with hydrogen, ending in a carboxyl group. The nature of these chains profoundly impacts the lipid's character. When every carbon atom in the chain is bonded to the maximum number of hydrogen atoms, with no double bonds between carbons, we call it a saturated fatty acid. These chains are straight and can pack tightly together, making the lipid solid at room temperature like butter. They are in a sense rigid and unyielding dancers, but introduce a double bond between carbon atoms, and the story changes. This creates a kink in the chain, preventing tight packing. These are unsaturated fatty acids, and lipids containing them, like olive oil, remain liquid at room temperature. They are the more fluid, free-spirited dancers, moving with a graceful bend. Triglycerides, whether saturated or unsaturated, are life's most efficient energy storage molecules, packing more than twice the energy per gram compared to carbohydrates. They are the long-term savings accounts of the cell, providing insulation and protection, a warm, protective embrace for our delicate internal machinery. Yet among the lipids, there is a truly remarkable architect, the phospholipid. Imagine a triglyceride, but with one of its fatty acid tails replaced by a phosphate group, often linked to another small polar molecule. This subtle alteration transforms the molecule into a creature of 2 worlds, a true amphipathic marvel. The phosphate-containing head is hydrophilic, drawn to water like a curious child reaching out. The 2 fatty acid tails, however, remain stubbornly hydrophobic, recoiling from water's embrace. This duality is not merely interesting, it is foundational to life itself. When phospholipids are placed in water, they spontaneously arrange themselves into a bilayer, or a double layer where the hydrophilic heads face outward toward the watery environment, and the hydrophobic tails tuck inward, away from the water, forming a secluded, oily core. This phospholipid bilayer is the very essence of the cell membrane, the delicate yet robust boundary that encloses every living cell, defining its internal world and regulating what passes through. It is a shimmering, dynamic fence, a silent guardian that allows life to maintain its distinct identity amidst the swirling chaos. And then we have the steroids yin, a class of lipids distinguished by their unique carbon skeleton. 4 fused rings. Cholesterol, often misunderstood, is a prime example. It is not just a molecule to be feared, it is a vital component of animal cell membranes, providing fluidity and stability. More profoundly, cholesterol serves as the precursor from which many other crucial steroids are synthesized, including our sex hormones like testosterone and estrogen. These steroids are the subtle messengers, the quiet whispers that orchestrate profound changes throughout an organism, guiding development and function. Lipids in their diverse forms are indeed the silent essential players, building the walls, fueling the engines, and carrying the messages that sustain life. Proteins, the master architects and dynamic workers. From the quiet elegance of lipids, we now soar into the realm of the proteins, the true workhorses and master architects of the cell. If lipids are the boundaries and energy stores, proteins are the dynamic performers, the intricate machinery and the structural scaffolding that give life its form, function, and incredible diversity. It is almost impossible to overstate their importance. Nearly every dynamic process within a living organism relies on proteins. The building blocks of these astonishing molecules are the amino acids. There are 20 common types, each a unique letter in the protein alphabet. Each amino acid shares a common core structure, a central carbon atom, affectionately called the alpha carbon, bonded to an amino group, NH2, a carboxyl group, COO, hydrogen atom, and a unique side chain, or R group. It is this R group, like a distinct personality trait, that gives each of the 20 amino acids its specific chemical properties. Whether it is hydrophilic or hydrophobic, acidic or basic, large or small, these subtle differences are the secret to protein diversity. Imagine these amino acids as a string of colourful, uniquely shaped beads. Proteins are formed when these amino acids link together in a specific sequence, head to tail, through a special type of covalent bond called a peptide bond. Again, this is a dehydration synthesis reaction, where the carboxyl group of one amino acid joins with the amino group of another, releasing a molecule of water. A chain of amino acids linked by peptide bonds is called a polypeptide. But a mere chain is not a functional protein. It is only the beginning of an astonishing transformation. The magic of proteins lies in their ability to fold into precise, intricate, three-dimensional shapes. This folding process is hierarchical, unfolding in 4 levels of structure. 1. Primary structure. This is the simplest level, yet the most fundamental. The unique, linear sequence of amino acids in the polypeptide chain. It's like the exact order of letters in a carefully written message. This sequence is determined by the genetic information encoded in DNA, and it is the blueprint for all subsequent folding. 2. Secondary structure. As the polypeptide chain elongates, local regions begin to fold into specific repeating patterns. These are primarily stabilized by hydrogen bonds forming between the backbone atoms, not the R groups, of nearby amino acids. The most common secondary structures are the elegant 1-alpha helix, a delicate spiral staircase, and the stately beta-pleated sheet, a folded accordion-like structure. These forms provide strength and flexibility. 3. Tertiary structure. This is where the overall complex three-dimensional shape of a single polypeptide chain emerges. This intricate folding is driven by a myriad of interactions between the R groups of distant amino acids. Hydrophobic R groups tend to cluster in the interior of the protein away from water, while hydrophilic R groups face outward. Hydrogen bonds, ionic bonds, and strong covalent or disulfide bridges, formed between two cysteine amino acids, all contribute to stabilizing this unique functional shape. This tertiary structure is absolutely critical. It is the specific contour, the precise arrangement of atoms that allows a protein to perform its particular job, whether it's binding to another molecule, catalyzing a reaction, or forming a structural element. 4. Quaternary structure. Not all proteins have this level, but many functional proteins are composed of 2 or more polypeptide chains, subunits, that come together to form a larger functional complex. Hemoglobin, the oxygen-carrying protein in our blood, is a perfect example, consisting of 4 separate polypeptide subunits precisely interlocked. The interactions holding these subunits together are similar to those found in tertiary structure. The profound truth about proteins is this: structure dictates function. A protein's specific intricate 3D shape is paramount to its ability to interact with other molecules and carry out its cellular role. If this delicate shape is disrupted by changes in temperature, pH, or exposure to certain chemicals, the protein can unfold, a process called denaturation. When a protein denatures, it loses its functional shape and consequently its biological activity. It's like a finely tuned musical instrument losing its harmony, or a precision tool losing its edge. Proteins perform an astounding array of functions. They act as enzymes, exquisitely specific catalysts that speed up biochemical reactions. They provide structural support, collagen in our skin, keratin in our hair. They provide transport as substances. Hemoglobin carries oxygen. Membrane proteins move ions. They serve as receptors for cellular communication. They facilitate movement, actin and myosin in muscle cells, and they even provide defense, antibodies fighting infection. Proteins are the ultimate shapeshifters and doers, the very embodiment of life's dynamic activity. Nucleic acids, the keepers of life's blueprint. Finally, we arrive at the very heart of life's information system, the nucleic acids. If proteins are the cellular workers and architects, then nucleic acids are the master blueprints, the ancient scrolls that contain all the instructions for building and operating an organism. They are the molecular repositories of heredity, ensuring that life's intricate dance continues from one generation to the next. The monomers of nucleic acids are called nucleotides. Each nucleotide is a complex 3-part molecule, like a tiny information-rich module. It consists of 1, a phosphate group, a phosphorus atom bonded to 4 oxygen atoms, 2, a 5-carbon sugar, either deoxyribose in DNA or ribose in RNA, 3, a nitrogenous base, a ring-shaped structure containing nitrogen. There are 5 main types of nitrogenous bases: adenine, A, guanine, G, cytosine C, thymine T, and uracil U. These individual nucleotides link together to form a long chain, a polynucleotide, through phosphodiester bonds that connect the sugar of one nucleotide to the phosphate of the next. This creates a sugar-phosphate backbone with the nitrogenous bases projecting outward, like a series of flags carrying specific symbols. There are two primary types of nucleic acids, each with a distinct and vital role. 1. Deoxyribonucleic acid DNA. This is the famed double helix, a magnificent spiraling ladder that forms the genetic material of all cellular life. In DNA, two polynucleotide strands intertwine, held together by hydrogen bonds forming between specific pairs of nitrogenous bases across the ladder's rungs. Adenine, A, always pairs with thymine, T, and guanine, G, always pairs with cytosine, C. This precise base pairing is crucial for DNA's ability to store and replicate genetic information faithfully. DNA is the stable long-term archive, the irreplaceable blueprint, carefully guarded within the nucleus of our cells. It is the wisdom passed down through eons, defining who we are and what we can become. 2. Ribonucleic acid, RNA. Unlike DNA, RNA is typically a single-stranded polynucleotide. While it also uses adenine, guanine, and cytosine, it substitutes uracil U for thymine T. RNA is far more versatile than DNA, playing multiple roles in the expression of genetic information. There are various types of RNA. Messenger RNA, mRNA, carries the genetic instructions from DNA to the protein-making machinery. Transfer RNA, or tRNA, brings the correct amino acids to build proteins, and ribosomal RNA, RNA, forms part of the ribosomes, the cellular factories where proteins are synthesized. RNA is the dynamic messenger, the skilled translator, the active participant in bringing the blueprint's instructions to life. Together, DNA and RNA orchestrate the fundamental flow of genetic information, often summarized as the central dogma of molecular biology. DNA's instructions are transcribed into RNA, and RNA's message is then translated into proteins. This elegant sequence from blueprint to worker is the very essence of how life builds, maintains, and perpetuates itself. We have now journeyed through the four great classes of biological macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Each, in its own unique way, contributes to the astonishing complexity and elegance of life. From the sweet energy of sugars to the boundary-forming lipids, from the dynamic shape-shifting proteins to the information-rich nucleic acids, these molecules interact in a symphony of unseen forces, building structures, catalyzing reactions, and transmitting the very essence of existence. As we prepare to move beyond these fundamental building blocks, let us carry this sense of profound wonder, knowing that these intricate components are merely the beginning of the story of how life, in all its forms, beautifully unfolds. We have journeyed far, haven't we, my curious friends? From the shimmering clouds of probability that define an atom to the elegant architecture of macromolecules, carbohydrates, lipids, proteins, and nucleic acids, we have meticulously unveiled the fundamental building blocks, the very letters and words of life's profound story. We've seen how atoms, like shy dancers, reach out to form bonds, how water orchestrates its vital roles, and how carbon, that magnificent architect, lays the foundation for life's grand designs. But now, a quiet whisper beckons us beyond the individual components. For these molecules, as wondrous as they are in isolation, do not exist in solitude. They are not merely scattered parts, they are participants in a much larger, more intricate ballet. Imagine, if you will, a vast symphony where each instrument, each note contributes to a harmonious whole. This is the realm we now enter, the breathtaking world of the cell. The cell, my dear explorers, is not just another structure. It is the fundamental unit of life itself. It is the smallest entity that can truly be called alive. It is a self-contained universe, a bustling microcosm where all the molecular dances we've observed thus far culminate in the miracle of existence. From the tiniest bacterium to the largest whale, from the delicate petal of a flower to the intricate neurons of our own minds, life expresses itself through that elegant simplicity and astonishing complexity of the cell. Think of it. Every breath we take, every thought we ponder, every beat of our heart is a testament to the ceaseless activity within countless cells. It is within these miniature worlds that the blueprint of DNA is read, the proteins are sculpted, and the energy of the universe is harnessed. It's a truth so profound it underpins all of biology. All living things are composed of cells. The cell is the basic unit of life, and all cells arise from pre-existing cells. These are not mere statements, they are the bedrock upon which our understanding of life is built, a testament to the continuous, unbroken chain of cellular existence stretching back billions of years. As we peer into this miniature world, we find that despite their immense diversity, all cells share a few universal features, like common threads woven through the tapestry of life. Every cell, without exception, is encased by a delicate yet formidable boundary, the plasma membrane. Ah, the plasma membrane. Do you remember our discussion of phospholipids, those remarkable molecules with their hydrophilic heads and hydrophobic tails? Here they truly shine. They arrange themselves into a magnificent woolly bilayer, a double layer that forms the very skin of the cell. This membrane is not a static wall, but a fluid dynamic mosaic, studded with a dazzling array of proteins. These proteins, our cellular workhorses, act as gates, channels, receptors, and identifiers, allowing the cell to communicate with its surroundings, to selectively import nutrients, and to export waste. The plasma membrane is the cell's sophisticated gatekeeper, a shimmering, intelligent boundary that maintains the cell's internal environment, a constant conversation between inside and out. It is a testament to the power of self-assembly, a marvel of molecular architecture that arises spontaneously from the very properties of its lipid and protein components. Within this protective embrace we find the cytoplasm, a bustling internal landscape. The fluid portion, the cytosol, is a jelly-like substance, a watery medium where countless biochemical reactions unfold, a vibrant internal sea teeming with dissolved ions, sugars, and of course our macromolecules. Suspended within this cytosol are the various organelles, the specialized miniature organs of the cell, which we shall explore shortly. And of course, every cell carries within it the sacred scroll of life, its genetic material, DNA. Whether housed within a dedicated chamber or simply coiled in a specific region, this nucleic acid, our blueprint, holds all the instructions for building and maintaining the cell. And to read these instructions, to translate them into the proteins that perform life's myriad functions, every cell possesses ribosomes. These tame molecular machines, composed of ribosomal RNA, rRNA, and proteins, are the cell's universal protein factories, tirelessly synthesizing the polypeptide chains that will fold into functional proteins. Now, with these universal features in mind, let's acknowledge a profound division in the cellular world, a distinction that separates life into two grand categories: prokaryotic cells and eukaryotic cells. Imagine for a moment the earliest forms of life on Earth. They were simple, elegant, incredibly resilient. These were the prokaryotes, a name meaning before nucleus. They are the pioneers, the ancient architects, and they continue to thrive in every corner of our planet, from the depths of the ocean to the most extreme environments. Prokaryotic cells like bacteria and archaea are characterized by their beautiful simplicity. They are typically smaller and less complex than their eukaryotic counterparts. Crucially, they lack a true nucleus. Their genetic material, a single circular chromosome, is coiled within a region called the nucleoid, but it is not enclosed by a membrane. Furthermore, prokaryotes do not possess membrane-bound organelles. Their cytoplasm is a single undivided space where all metabolic activities occur. Despite their apparent simplicity, they are exquisitely efficient, capable of astonishing metabolic diversity, and absolutely essential for the health of our planet. They often possess a rigid cell wall outside their plasma membrane, providing structural support and protection, and some may have flagella for movement or pili for attachment. Then there are the eukaryotic cells, meaning true nucleus. These are the cells of animals, plants, fungi, and protists, the cells that make up you and me. Eukaryotes represent a leap in cellular complexity, a grand evolutionary innovation. They are generally larger than prokaryotes and, most significantly, possess a true membrane-enclosed nucleus that houses their genetic material. But the marvel doesn't stop there. Eukaryotic cells are a symphony of internal compartments, an intricate network of some membrane-bound organelles, each performing specialized tasks. This compartmentalization is a game-changer, allowing eukaryotic cells to carry out a vast array of complex biochemical reactions simultaneously, with efficiency and precision, much like different departments in a well-organized factory. Let's take a gentle stroll through the bustling interior of a typical eukaryotic cell, a guided tour of its magnificent architecture. Our first stop inevitably is the nucleus, the cell's command center, its library, its very heart. Enclosed by a double membrane called the nuclear envelope, which is perforated by tiny pores to regulate molecular traffic, the nucleus is where the cell's precious DNA resides, organized into structures called chromatin. It is here that the genetic blueprint is protected, replicated, and transcribed into RNA. Within the nucleus, we also find the nucleolus, a dense region where ribosomal RNA, rRNA, is synthesized and assembled with proteins to form ribosomal subunits, the very components of our protein factories. Radiating from the nuclear envelope, we encounter a vast interconnected network of membranes known as the endomembrane system. This system is a testament to the cell's internal communication and transport capabilities, a coordinated effort to synthesize, modify, package, and transport lipids and proteins. First, the endoplasmic reticulum, ER. A labyrinthine network of membranes that extends throughout the cytoplasm. We speak of two types. The rough ER, so named because its surface is studded with ribosomes, is the site where proteins destined for secretion, insertion into membranes, or delivery to other organelles are synthesized and folded. It's like the cell's initial protein processing plant, where new polypeptide chains begin their journey, often aided by the molecular chaperones, before being modified and sent on their way. The smooth ER, which lacks ribosomes, is involved in a different set of crucial tasks. The synthesis of lipids, including phospholipids and steroids, detoxification of drugs and poisons, especially abundant in liver cells, and storage of calcium ions. It's the cell's versatile workshop for lipid production and internal regulation. From the ER, many proteins and lipids travel to the Golgi apparatus, often described as the cell's post office or shipping center. This organelle consists of flattened membranous sacs called cisternae, arranged in stacks. Here, molecules are further modified, sorted, and packaged into vesicles, tiny membrane-bound sacs, for transport to their final destinations, whether within the cell or for secretion outside. The Golgi is a master of molecular logistics, ensuring that each molecule arrives precisely where it is needed. Other components of the endomembrane system include lysosomes, the cell's recycling centers. These are membrane-bound sacs containing powerful hydrolytic enzymes that digest macromolecules, old organelles, and foreign particles, breaking them down into their molecular components for reuse. In plant and fungal cells, large central wall vacuoles play similar roles, along with storing water, nutrients, and waste products, and maintaining turgor pressure. Stepping outside the endomembrane system, we encounter the cell's remarkable energy converters. The mitochondria, often called the powerhouses of the cell, are ubiquitous in nearly all eukaryotic cells. These fascinating organelles, with their distinctive double membranes and folded inner cristae, are where cellular respiration occurs, the process that generates the vast majority of the cell's ATP, its primary energy currency, from sugars and other organic fuels. They are the engines that drive all cellular activity, the silent generators of life's energy. In plant and algal cells, we find another magnificent energy converter, the chloroplasts. These are the solar panels of the living world, the sites of photosynthesis, where sunlight energy is captured and transformed into chemical energy, creating sugars from carbon dioxide and water. Chloroplasts, like mitochondria, possess their own unique DNA and ribosomes, hinting at their ancient origins as independent prokaryotic cells that were engulfed by early eukaryotes. A profound story of symbiosis that reshaped the course of life on Earth. Finally, providing shape, support, and facilitating movement within the cell is the cytoskeleton. This intricate network of protein filaments, microtubules, microfilaments, and intermediate filaments acts as the cell's internal scaffolding, its highway system, and its dynamic muscle. It helps maintain cell shape, anchors organelles, and enables cell motility, from the crawling of an amoeba to the precise movements of chromosomes during cell division. Here again we see the profound importance of proteins, acting as the very architects and engineers of cellular form and function. So you see, my friends, the cell is not merely a collection of molecules, it is a meticulously organized, breathtakingly dynamic entity, a testament to the power of molecular self-assembly and the exquisite precision of biological processes. Each macromolecule we explored in the previous chapters finds its purpose, its stage, within the confines of this living theatre. From the lipid bilayer of the membrane to the protein enzymes orchestrating reactions, from the DNA blueprint in the nucleus to the carbohydrate fuel, every component plays a vital role in the grand, continuous performance of life. As we conclude this chapter, I invite you to pause and simply marvel. Marvel at the sheer ingenuity, the inherent beauty, and the profound interconnectedness that defines every single cell. It is a universe within a universe, a silent, ceaseless dance that defines existence itself. And with this understanding of the cellular landscape, we are now truly ready to explore the dynamic processes that animate these magnificent structures, to witness the very essence of life in action. We have journeyed together, haven't we, through the intricate architecture of the cell, marvelling at its elegant design, its specialized compartments, and the silent dignity of its many organelles. We've peered into the nucleus, watched the endoplasmic reticulum at its tireless work, and understood the Golgi's meticulous packaging. But a magnificent cathedral, however grand its design, is truly alive only when the faithful gather within its walls, when whispers echo and light streams through stained glass, illuminating movement and purpose. So it is with the cell. Its structure, however perfect, is but the stage. Now, let us draw back the curtain and witness the hidden ballet, the ceaseless, vibrant activity that animates these microscopic worlds. For a cell is not a static blueprint. It is a bustling, dynamic city, a miniature universe teeming with chemical transformations. This vast, intricate network of chemical reactions, the very essence of life's continuous unfolding, we call metabolism. Imagine, if you will, a grand symphony, constantly playing. Every note, every instrument, every subtle shift in tempo contributes to the whole. Metabolism is precisely this, a symphony of countless chemical reactions, each one perfectly timed, perfectly orchestrated to sustain the living state. It is the sum of all the chemical processes that occur within a living organism, a delicate balance between building up and breaking down, creating and consuming. We can broadly divide this grand symphony into two fundamental movements. The first movement, anabolism, is the constructive phase. Here, smaller, simpler molecules are painstakingly assembled into larger, more complex ones. Think of it as the cell's construction crew, building proteins from amino acids, or complex carbohydrates from simple sugars, or even entire new cellular components. This building process, as you might intuitively grasp, requires an input of energy. Much like constructing a magnificent building demands resources and effort. The second movement, catabolism, is the deconstructive phase. In this part of the symphony, larger, more complex molecules are broken down into smaller, simpler ones. This is where the cell dismantles old structures, or more importantly, extracts the precious energy stored within the chemical bonds of nutrient molecules. When we digest our food, for instance, our cells are engaged in catabolism, breaking down starches into glucose or proteins into amino acids. Crucially, these catabolic reactions release energy, much like dismantling an old structure can salvage valuable materials. So we see a beautiful rhythmic dance, anabolism building, catabolism breaking, and in this eternal cycle, energy is both consumed and liberated. But how does this energy, once released, become usable by the cell? How does the cell pay for its anabolic construction projects, or the countless other tasks it must perform? Here, we encounter the universal currency of life, ATP, or adenosine triphosphate. Imagine ATP as a tiny shimmering coin, minted in the very furnaces of life, ready to be exchanged for a moment of movement, a flicker of thought, a beat of a heart, or the precise assembly of a new protein. It is the direct and immediate source of energy for almost all cellular activities. Let's gently unravel the nature of this remarkable molecule. ATP is a nucleotide much like the building blocks of DNA and RNA we discussed, but with a crucial difference. It consists of three parts: a nitrogenous base called adenine, a 5-carbon sugar called ribose, and, most importantly for our current discussion, a chain of 3 phosphate groups. The magic, the very power of ATP, resides in the bonds connecting these phosphate groups. Specifically, the bond between the second and third phosphate group, and to a lesser extent the one between the first and second, are often referred to as high-energy bonds. While not truly high energy in a universal sense, they are readily broken by hydrolysis, releasing a significant amount of usable energy in the process. When a cell needs energy, perhaps to pump ions across a membrane, to contract a muscle fibre, or to synthesize a new molecule, it simply spends an ATP coin. A water molecule cleaves off the terminal phosphate group, transforming ATP into ADP, adenosine diphosphate, and an inorganic phosphate group, and voila, energy is liberated. This energy is then immediately harnessed to power the cellular work. But life is a continuous process, and these ATP coins are spent rapidly. A typical human body cycles through its entire body weight in ATP every single day. This means ATP must be constantly regenerated. Catabolic reactions, breaking down nutrients like glucose, provide the energy to reattach that third phosphate group to ADP, turning it back into ATP, ready to be spent again. It's a breathtakingly efficient, ceaseless cycle of energy transfer. Now imagine our metabolic symphony, vast and complex. How do all these hundreds, even thousands of distinct chemical reactions occur at the precise right moment, at the perfect speed, and with such exquisite specificity within the gentle confines of a living cell? The temperatures and pressures within a cell are mild. They are not the fiery furnaces of an industrial chemical plant. If left to their own devices, many vital biochemical reactions would proceed far too slowly to sustain life, or they might not happen at all. This is where the true maestros of the cellular symphony enter, the enzymes. If metabolism is the grand performance, then enzymes are the dedicated, highly skilled conductors, ensuring each note is played at the perfect tempo, each instrument brought in at the right moment, guiding the flow of energy and matter with unparalleled precision. What exactly are enzymes? For the most part, they are magnificent protein molecules, intricately folded into specific three-dimensional shapes. Their primary role is to act as biological catalysts. A catalyst in general chemistry is a substance that speeds up the rate of a chemical reaction without being consumed in the process. Enzymes do precisely this, but with an elegance and the efficiency that synthetic catalysts can only dream of. Think of a chemical reaction needing to occur, perhaps two molecules needing to join together, or a large molecule needing to be broken apart. Often there's an energy barrier, like a small hill or even a mountain pass, that the reactants must overcome before the reaction can proceed. This is called the activation energy core. It's the initial input of energy required to get the reaction started, even if the overall reaction releases energy. For many vital reactions, this activation energy is simply too high to be overcome efficiently at body temperature. Enzymes, in their wisdom, don't change the overall energy difference between the reactants and products. Instead, they gently and precisely lower the activation energy. Imagine an enzyme as an architect designing a tunnel through that mountain pass, or a ramp over that small hill, making the journey far easier and faster for the molecules involved. They provide an alternative lower energy pathway for the reaction to occur. The key to an enzyme's extraordinary power lies in its specificity. Each enzyme is typically designed to catalyze only one or a very small number of specific reactions. How does it achieve this? Each enzyme possesses a unique three-dimensional region called the active site. This active site is like a perfectly shaped lock complementary to the shape of its specific reactant molecule, which we call the substrate. When the substrate molecule encounters its specific enzyme, It fits precisely into the active site, much like a key into a lock, forming an enzyme-substrate complex. This interaction isn't always rigid. Sometimes the enzyme might subtly change its shape to better embrace the substrate, a concept known as the induced fit model, like a gentle, custom-tailored glove moulding around a hand. Once nestled in the active site, the enzyme subtly strains the bonds within the substrate, or brings reacting molecules into optimal proximity, facilitating the chemical transformation. After the reaction occurs, the products are released, and the enzyme emerges unchanged, ready to bind to another substrate molecule and catalyze the reaction again and again and again. It is truly tireless in its work. The efficiency of enzymes is astounding. A single enzyme molecule can catalyze thousands, even millions of reactions per second. And their activity is exquisitely regulated, ensuring the cell's metabolic needs are met precisely. This regulation is influenced by various factors. The gentle warmth of our bodies, for instance, provides an optimal temperature for most human enzymes to function. Too cold and they slow down, too hot and their delicate three-dimensional structure can unravel, a process called aldenaturation, leading to a loss of function, much like a finely tuned instrument losing its shape and thus its ability to play. Similarly, a specific sharp pH range is crucial for optimal enzyme activity, as extreme acidity or alkalinity can disrupt the enzyme's structure. The concentration of both enzyme and substrate also plays a role, influencing how frequently they encounter each other. Sometimes enzymes also require helper molecules called permeable cofactors, inorganic ions like zinc or iron, or coenzymes, organic molecules often derived from vitamins, to assist in their catalytic duties. So we see the profound interconnectedness. Catabolic reactions break down molecules, releasing energy, much of which is captured in the form of ATP. Anabolic reactions then consume this ATP to build new molecules requiring energy, and orchestrating this entire magnificent metabolic symphony, ensuring every reaction occurs at the right time and with the right speed, are the tireless, precise, and beautiful enzymes. This ceaseless flow of energy and matter, guided by the elegant hand of enzymes, is the very pulse of life within every cell. It is a testament to the intricate beauty and profound order that underlies all biological existence. As we contemplate this dynamic interplay, we begin to truly appreciate that the cell is not merely a collection of parts, but a living, breathing, endlessly transforming entity, a universe in miniature constantly performing its vital hidden dance. What wonder, what profound artistry we discover at this fundamental level. Let us carry this sense of awe as we delve deeper into the secrets of life's intricate mechanisms. As we delve deeper into the breathtaking intricacies of life, we find ourselves ever more enchanted by the delicate balance and profound efficiency that governs the molecular world. In our last gathering, we marveled at the tireless work of enzymes, those masterful biological catalysts that orchestrate the myriad chemical reactions essential for existence, gently nudging them forward, lowering the barriers of activation energy, and ensuring life's symphony plays on without discord. We understood how ATP, the shimmering coin of cellular energy, fuels these countless metabolic dances, a universal currency accepted in every corner of the living realm. But a question perhaps lingers in the quiet chambers of your mind, a most fundamental query. Where does this precious ATP come from? How do cells, these minuscule architects of life, generate the energy they need to build, to break down, to move, to simply be? Today, my dear explorer, we embark on a journey into the grandest energy cycles of our planet, a tale of sunlight woven into substance, and of substance carefully dismantled to release that stored radiance. we shall explore the twin pillars of cellular energy, cellular respiration, and photosynthesis. Together, they form a cosmic breath, an exquisite exchange that sustains nearly all life on Earth. Imagine with me, if you will, the gentle hum of a hidden engine, endlessly working within each of your cells, within every leaf, every bacterium. This is not a roaring furnace, but a series of meticulously controlled whispers, a cascade of tiny, purposeful steps designed to extract energy with remarkable precision. Let us begin with cellular respiration, the process by which cells unlock the energy stored within organic molecules, primarily glucose, to forge those precious ATP molecules. It is in essence a controlled burning of fuel, but unlike the wild, destructive flame that consumes a log in a bonfire, cellular respiration is a slow, elegant dance, extracting energy step by deliberate step, minimizing waste and maximizing yield. This profound journey begins in the cytoplasm, the jelly-like substance filling your cells with a process we call the glycolysis. The very name glycolysis whispers its secret: sugar splitting. Here, a single molecule of glucose, that 6-carbon sugar we've come to know, is gently coaxed and twisted through a series of 10 enzymatic steps into 2 smaller 3-carbon molecules called pyruvate. It's an ancient pathway, a testament to life's earliest beginnings, so fundamental that it occurs in nearly all living organisms, from the smallest bacterium to the largest whale. In this initial phase, a modest offering of ATP is produced, a small down payment on the energy to come, along with some electron carriers, molecules that will ferry high-energy electrons to later stages. Remarkably, glycolysis does not require oxygen. It is an anaerobic dance, capable of proceeding even in the absence of that vital gas. But if oxygen is present, as it is in most of our cells, the pyruvate molecules embark on a deeper journey, venturing into the very heart of the cell's powerhouses, the mitochondria. The mitochondria, you remember them, don't you? Those bean-shaped organelles, often called the power plants of the cell. Here, a symphony of reactions unfolds across their inner and outer membranes, a truly magnificent display of molecular architecture and function. First, each pyruvate molecule undergoes a transformation, losing a carbon atom released as carbon dioxide, which you exhale with every breath, and becoming a 2-carbon compound called acetyl-CoA. This acetyl-CoA then gracefully enters the citric acid cycle, also known as the Krebs cycle, a beautifully cyclical series of reactions that further dismantles the original glucose molecule. Imagine a tiny molecular Ferris wheel, where carbon atoms are loaded on, spun around, and then released as more carbon dioxide. With each turn of this exquisite cycle, more electron carriers, NADH and FADH2, are generated, becoming increasingly laden with high-energy electrons, ready for the final, most dramatic act of cellular respiration. A little more ATP is also produced here, but the true treasure lies in those electron carriers. And now, for the grand finale, the stage where the vast majority of ATP is forged, oxidative phosphorylation. This process unfolds across the inner membrane of the mitochondrion, a membrane intricately folded into cristae, maximizing its surface area for this crucial work. Here, the electron carriers, NADH and FADH2, arrive, bearing their precious cargo of high-energy electrons. These electrons are then passed down a series of protein complexes embedded within the membrane, much like a tiny molecular cascade or a series of miniature waterfalls. This is the electron transport chain. As the electrons fall from one protein complex to the next, they release small bursts of energy at each step. This energy is not wasted. It is ingeniously harnessed to pump protons, hydrogen ions, from the inner compartment of the mitochondrion into the intermembrane space, creating a high concentration of protons there. Think of it like building a dam, creating a powerful reservoir of water. This proton gradient, this difference in concentration, represents stored potential energy, a magnificent tension waiting to be released. And what releases it? A remarkable molecular machine called ATP synthase. This enzyme, a true marvel of biological engineering, is embedded in the mitochondrial inner membrane, acting like a tiny turbine. As protons flow back down their concentration gradient, rushing through ATP synthase, like water rushing through a turbine, they cause the enzyme to spin, quite literally, synthesizing ATP from ADP and inorganic phosphate. This elegant process, the coupling of electron transport with ATP synthesis via a proton gradient, is known as chemiosmosis. It is here, in this final breathtaking stage, that the vast majority of ATP, the very energy currency of life, is meticulously crafted. And what happens to those electrons after their energetic journey down the chain? They are finally accepted by oxygen, which then combines with protons to form water. This, my dear reader, is why we breathe. Oxygen is not merely a gas. It is the vital electron acceptor, the final destination for the weary electrons, without which the entire exquisite machinery of oxidative phosphorylation would grind to a halt. But what if oxygen is scarce? What if our cells, or those of other organisms, find themselves in an environment devoid of this crucial element? Life, ever resourceful, has an alternative: anaerobic respiration or fermentation. In these conditions, glycolysis can still proceed, producing a small amount of ATP. However, without oxygen to accept electrons, the electron transport chain cannot function, and the electron carriers, NADH, cannot be regenerated. To keep glycolysis going, pyruvate is converted into other products like lactate in our muscle cells during intense exercise, or ethanol by yeast in the making of bread and beer. These processes regenerate the electron carriers needed for glycolysis, allowing a modest, albeit much less efficient production of ATP to continue, a testament to life's tenacity. Now, having explored how cells extract energy, let us turn our gaze to the ultimate source of nearly all life's energy, the sun itself. Let us journey into the realm of photosynthesis, the miraculous process by which light energy is captured and transformed into the chemical energy stored in organic molecules like glucose. If cellular respiration is the controlled burning of fuel, photosynthesis is the ultimate alchemy, turning light into substance, weaving the very fabric of life from thin air and pure radiance. This profound transformation occurs primarily in plants, algae, and some bacteria, within specialized organelles called chloroplasts. You may recall them as the green disc-shaped structures, the solar panels of the plant cell. Photosynthesis, too, is a two-stage masterpiece. The first stage, the cell-light-dependent reactions, are precisely what their name implies. They require light. These reactions unfold within the thylakoid membranes inside the chloroplast, which are stacked into structures called grana. Here, chlorophyll, the vibrant green pigment that gives plants their color, acts as a molecular antenna, greedily absorbing photons of sunlight. When a chlorophyll molecule captures light energy, it excites electrons to a higher energy level. These energized electrons are then passed down an electron transport chain, remarkably similar in principle to the one we saw in mitochondria, though operating in reverse, building energy rather than releasing it. As these electrons move down the chain, their energy is used to pump protons across the thylakoid membrane, once again creating a proton gradient. And just as in cellular respiration, this proton gradient drives ATP synthase, producing ATP. Simultaneously, water molecules are split, providing the electrons needed to replenish the chlorophyll and releasing oxygen gas as a byproduct. Yes, my friends, the very oxygen we breathe, the life-giving breath of our planet, is a graceful gift from the light-dependent reactions of photosynthesis. At the end of this chain, the high-energy electrons are also used to reduce another electron carrier, NADP+, into NADPH, another form of stored chemical energy. The ATP and NADPH, brimming with captured light energy, are now ready for the second stage, the light-independent reactions, more commonly known as the Calvin cycle. These reactions occur in the stroma, the fluid-filled space surrounding the thylakoids within the chloroplast, and they do not directly require light. However, they are absolutely dependent on the products of the light reactions. The Calvin cycle is a truly elegant sequence of steps where carbon dioxide from the atmosphere is fixed, that is, it is incorporated into existing organic molecules. Imagine the chloroplast as a master chef taking simple carbon dioxide molecules and using the energy from ATP and the reducing power of NADPH, our energy carriers from the light reactions, to meticulously assemble them into larger, more complex sugar molecules, eventually forming glucose. It's a wondrous feat of molecular construction, turning gas into sustenance, energy into matter, and so we complete a magnificent circle. Photosynthesis, fueled by the sun's embrace, creates the glucose and oxygen that cellular respiration so efficiently uses. In turn, cellular respiration releases the carbon dioxide and water that photosynthesis eagerly reclaims. It is an exquisite planetary dance, a ceaseless exchange of energy and matter, binding all life into an interdependent web. The breath of the forest feeds the breath of the animal, and the exhale of the animal nourishes the forest. You see, my curious companions, life at its most fundamental level is an intricate tapestry of energy transformation. From the gentle splitting of sugar in glycolysis to the powerful cascade of the electron transport chain, from the sun's kiss on a chlorophyll molecule to the meticulous construction of glucose in the Calvin cycle, every step is a testament to nature's profound ingenuity. We are not merely observing these processes, we are witnessing the very pulse of life, the hidden engines that power every thought, every movement, every silent growth. Let us carry this wonder with us, ever mindful of the beautiful unseen forces that sustain our world. Having witnessed the magnificent interconnected dance of energy that sustains all life, the graceful exchange between the sun-drenched chloroplasts and the tireless mitochondria, let us now turn our gaze inward to the very life cycle of a cell itself. For what good is a perfect energy system if the cell cannot grow, cannot repair, cannot multiply? Here we discover another profound rhythm, a silent, intricate ballet that ensures the continuity of existence: cell division. Imagine, if you will, a single cell, a miniature universe pulsating with purpose. It breathes, it metabolizes, it performs its specialized tasks with unwavering dedication. But no cell lives forever in isolation, nor can an organism grow from a single spark. From the moment of conception, through growth, repair, and even the constant renewal of our own tissues, the symphony of life relies on cells making more of themselves. This isn't just about simple multiplication, it's about the faithful transmission of life's most precious blueprint, the genetic code, from one generation of cells to the next. We speak of the cell cycle, not merely as a moment of division, but as the entire journey a cell undertakes from its birth until it too divides into two daughter cells. It's a continuum, a loop of growth, preparation, and ultimate reproduction, orchestrated with a precision that borders on the miraculous. This cycle is broadly divided into two main phases. Interphase, the period of growth and preparation, and the M phase, mitotic phase, where the cell finally divides. Let us gently step into interphase, which, despite its name suggesting an in-between state, is anything but dormant. This is where the cell truly lives, breathes, and prepares for its monumental task. Interphase itself is composed of three distinct subphases, G1, S and G2. The first act is the G1 phase, or gap 1. Think of this as the cell's main stage for daily living. Here, the cell grows in size, synthesizes proteins and organelles, and carries out its specific metabolic functions. A liver cell detoxifies, a muscle cell contracts, a neuron transmits signals, all while growing and assessing its environment. It's a period of active life where the cell decides whether the conditions are right to proceed with division. We might imagine it as a thoughtful pause, a careful assessment of resources and readiness. Following a successful G1, the cell enters the pivotal S phase, the synthesis phase. This is the heart of preparation, where the cell's entire genetic library, its precious DNA, is meticulously copied. Imagine a scribe working with utmost care and precision, duplicating every single page of an ancient sacred text, ensuring not a single letter is out of place. This process, known as DNA replication, is semi-conservative. Each new DNA molecule consists of one original strand and one newly synthesized strand. It is a testament to nature's elegance, ensuring fidelity as the helix unwinds and new nucleotides are added, guided by the template strand. By the end of the S phase, each chromosome, which was once a single thread, now consists of two identical copies called sister chromatids, joined together at a central point called the centromere. They are still considered one chromosome at this stage, but they are poised for separation, like two inseparable twins holding hands. After the arduous task of DNA replication, the cell enters G2 phase, or gap 2. This is the final sprint before division. The cell continues to grow, synthesizes additional proteins and organelles, and, crucially, checks its work. It's a quality control checkpoint, ensuring that all the DNA has been replicated accurately and that the cell is truly ready to embark on the complex process of division. Think of it as a final rehearsal, a last-minute check of costumes and props before the curtain rises on the M phase. Now, with all preparations complete, we arrive at the spectacular M phase, the mitotic phase. This is where the physical act of cell division occurs, typically encompassing two distinct yet integrated processes, mitosis and cytokinesis. Mitosis, specifically, refers to the division of the nucleus and its genetic material, ensuring that each new daughter cell receives an identical set of chromosomes. This is the cornerstone of growth, tissue repair, and asexual reproduction in many organisms. Let us observe the intricate dance of mitosis, a four-act play of profound beauty and precision. The first act is prophase, As the curtain rises, a remarkable transformation begins within the nucleus. The sprawling diffuse chromatin, which was a tangled mess of DNA and proteins during interphase, starts to coil and condense, becoming tightly packed and visible under a microscope as discrete chromosomes. These are our sister chromatids, still joined at their centromeres, looking like tiny X-shapes. Simultaneously outside the nucleus, the mitotic spindle begins to form. This intricate network of microtubules, like delicate scaffolding, will guide the chromosomes to their proper places. The nuclear envelope, the protective membrane surrounding the nucleus, also begins to break down, dissolving like a morning mist, allowing the spindle fibers to interact with the chromosomes. Next, we move to metaphase. A moment of exquisite alignment. The condensed chromosomes, now fully formed and distinct, migrate to the cell's equator, forming a perfect line along what we call the metaphase plate. Imagine a group of dancers, each pair of sister chromatids, taking their precise positions on a stage, waiting for their cue. Each sister chromatid is firmly attached to the spindle fibers originating from opposite poles of the cell, ensuring that when the time comes, they will be pulled apart equally. It is a moment of poised stillness, a breathtaking tableau before the grand separation. The third act, anaphase, is swift and dramatic. Suddenly, the centromeres that held the sister chromatids together cleave, and the identical sister chromatids are pulled apart. They're no longer called sister chromatids, but are now considered individual chromosomes, once again. The spindle fibers, like tiny, powerful ropes, shorten, pulling these newly separated chromosomes towards opposite poles of the cell. Each pole receives a complete, identical set of genetic information. It is a meticulously choreographed exodus, ensuring that each future daughter cell will inherit the exact same genetic blueprint. Finally, we arrive at telophase, the concluding act of nuclear division. As the chromosomes reach their respective poles, they begin to decondense, unwinding from their tightly packed forms back into diffuse chromatin. New nuclear envelopes begin to reform around each set of chromosomes, creating two distinct nuclei within the single still dividing cell. The spindle fibers disassemble, their work complete. The cell briefly appears to have two nuclei, each a mirror image of the other, preparing for their independent lives. But the story isn't quite over. Mitosis, the division of the nucleus, is followed by our cytokinesis, the division of the cytoplasm. This is the physical partitioning of the cell into two distinct independent daughter cells. In animal cells, a cleavage furrow forms a contractile ring of actin and myosin filaments that pinches the cell in two, much like tightening a drawstring around a balloon. For plant cells, with their rigid cell walls, the process is different. A cell plate forms in the middle, growing outward from the center until it fuses with the existing plasma membrane and cell wall, effectively building a new wall to separate the two daughter cells. And there you have it. Two brand new cells, genetically identical to the parent cell, ready to embark on their own journey through the cell cycle. This meticulous process of mitosis ensures the faithful transmission of genetic information, allowing for growth from a single fertilized egg into a complex multicellular organism for the replacement of old or damaged cells and for the asexual reproduction of countless life forms. This entire cycle from G1 through S, G2, and finally M phase is not a chaotic free-for-all. Oh no, it is under incredibly tight control. Imagine a series of vigilant checkpoints like wise guardians stationed at critical junctures, ensuring that the cell is healthy, its DNA intact, and all conditions favourable before allowing it to proceed to the next stage. These checkpoints are regulated by complex molecular machinery involving proteins like cyclins and cyclin-dependent kinases, CDKs. It is a testament to the cell's inherent wisdom, a built-in regulatory system designed to prevent errors. For when this delicate control is lost, when cells disregard the checkpoints and divide uncontrollably, we encounter the devastating phenomenon we call cancer. Yet, as remarkable as mitosis is, it presents only one facet of cellular reproduction. While it ensures genetic fidelity, there is another, equally profound mode of division, one that introduces the beautiful element of variation. For the purposes of sexual reproduction, for the creation of new individuals from two parents, a different kind of cellular dance is required, one that reduces the genetic material by half and shuffles the deck of life's possibilities. But that, my curious friend, is a story for our next exploration, where we will delve into the wonders of meiosis and the genesis of genetic diversity. For now, let us simply marvel at the precision, the elegance, and the sheer continuity that the cell cycle, particularly mitosis, bestows upon the living world. And so we arrive, dear companion, at another threshold of understanding, a new chamber in our dreamy academy. Last we met, we gazed upon the precise, almost mathematical ballet of mitosis, a process designed for faithful replication, for the perpetuation of sameness. It was a testament to order, to the meticulous copying of life's blueprint. But life, as we know, is also a symphony of change, a vibrant canvas painted with endless variations. What if the goal isn't sameness, but rather a deliberate, exquisite shuffling of the deck? What if the very engine of continuity also held the secret to endless novelty? Today we turn our gaze to another, equally profound mode of cellular division, one that introduces the beautiful element of variation, a process vital for the purposes of sexual reproduction. Let us gently step into the realm of meiosis, the exquisite dance that sculpts the very fabric of inherited diversity. Imagine, if you will, that the story of life isn't just about making perfect copies of a beloved book. Sometimes it's about creating an entirely new volume, one that draws inspiration from two different, cherished libraries blending their tales and characters in ways never before seen. This is the essence of meiosis, a division not for growth or repair, but for the creation of unique reproductive cells, known as gametes, sperm and egg. You see, in most of the cells we've discussed so far, the somatic cells of your body, chromosomes exist in pairs. One set of chromosomes comes from your biological mother, and the other set from your biological father. These pairs are called homologous chromosomes. They carry genes for the same traits at the same locations, though the specific versions, alleles, of those genes might differ. If you have, say, 46 chromosomes in a human somatic cell, you actually have 23 pairs. We refer to such cells as diploid, a state elegantly denoted as kun, signifying two sets of chromosomes. Now, if two diploid cells were to simply fuse, the resulting offspring would have double the normal chromosome number, an unsustainable genetic overload. Nature, in its boundless wisdom, devised a clever solution: meiosis. This specialized division reduces the chromosome number by half, creating cells that are haploid, containing only one set of chromosomes denoted as. These haploid cells are our gametes. When a haploid sperm fuses with a haploid egg during fertilization, the diploid state is beautifully restored and a new unique individual begins its journey. Meiosis is not a single act, but a two-part epic, a grand performance divided into meiosis Y and meiosis II. Each part has its own distinct purpose, its own unique choreography. Let us first immerse ourselves in the quiet unfolding of meiosis I, often referred to as the reductional division. Here, the primary goal is to separate those homologous chromosome pairs, to gently part the genetic partners that have journeyed through life side by side. It begins, as all grand narratives do, with a preparation phase, a prophase I, that is perhaps the most intricate and profound phase of all meiosis. While similar to mitotic prophase in some ways, the nuclear envelope dissipates, the spindle fibers begin to form, it holds a secret, a profound event that is the very heart of genetic variation. In this sacred space of prophase I, the homologous chromosomes, those pairs we just spoke of, find one another. They don't just exist in the same cell, they actively seek each other out, aligning themselves precisely gene for gene in a process called synapsis. Imagine two long, intricate tapestries, each woven with threads of genetic information, carefully laying themselves side by side, their patterns mirroring one another. As they align, they form a structure known as a tetrad, a group of 4 chromatids. Remember, each chromosome has already duplicated, so it consists of 2 sister chromatids. And here, in this intimate embrace, occurs the magical, transformative event, crossing over. It is as if the homologous chromosomes, in their close proximity, exchange fragments of their genetic material. Picture those tapestries we imagined earlier. Now, visualize small sections of thread from one tapestry carefully unweaving and reweaving themselves into the other, and vice versa. This physical exchange creates entirely new combinations of alleles on each chromatid. A chromosome that once carried only maternal traits on a certain segment might now carry a blend of maternal and paternal traits. This reshuffling is utterly random, exquisitely precise, and fundamentally unique for every meiotic event. It is the first, most powerful engine of genetic diversity, ensuring that no 2 gametes are ever truly identical, even from the same individual. The stories whispered between these chromosomes are never quite the same. Following this profound intermingling, we move to metaphase I. The homologous pairs, now intertwined with their newly exchanged segments, migrate to the cell's equator, forming the metaphase plate. But here's another twist in the dance. The orientation of each homologous pair at the metaphase plate is entirely random and independent of the others. This is known as independent assortment. Think of it like a line of dancers, each pair facing either left or right before the music begins. The direction one pair faces has no bearing on the direction any other pair faces. This random alignment means that when the pairs separate, the resulting daughter cells will receive a completely unpredictable mix of maternal and paternal chromosomes. Another layer of unique genetic combination is woven into the tapestry of life. Then comes anaphase Y, the moment of separation. Unlike mitosis, where sister chromatids pull apart in meiosis Y, and it is the homologous chromosomes that separate. One full chromosome, still composed of 2 sister chromatids from each pair, moves towards opposite poles of the cell. The partners part ways, but each still holds on to its duplicated self. Finally, telophase Y and Yika, cytokinesis, conclude the first grand act. The chromosomes arrive at the poles, the nuclear envelope may briefly reform, and the cell divides into 2 new cells. Crucially, each of these 2 daughter cells is now haploid N in terms of chromosome and number, because the homologous pairs have separated, but each chromosome still consists of two sister chromatids. This means the DNA content is still duplicated, but the genetic complement is distinct and reduced. A brief, often subtle interlude, sometimes called interkinesis, may occur, a pause before the next act. There is no further DNA replication. The genetic material is already prepared. Now we enter meiosis II, often referred to as the equational division. This phase bears a striking resemblance to mitosis, but with a critical difference. It begins with the two haploid cells produced in meiosis I, not a diploid cell. Its purpose is to separate the sister chromatids, much like in mitosis. In Hoc prophase II, the nuclear envelope, if reformed, dissolves again, and the spindle apparatus reforms in each of the two haploid cells. During metaphase II, the chromosomes, each still made of two sister chromatids, align individually along the metaphase plate of each cell. Anaphase II witnesses the final separation. The sister chromatids finally pull apart, moving to opposite poles. They are now considered individual chromosomes. And finally, telophase II and cytokinesis bring the entire magnificent process to a close. Nuclear envelopes reform around the separated chromosomes at each pole. The cells divide and the result is four distinct genetically unique haploid N daughter cells. These are our precious gametes, ready for their role in the continuation of life. Let us pause for a moment, dear friend, and reflect on the profound sources of genetic variation that meiosis so elegantly orchestrates. We've touched upon them, but let's bring them together like the threads of a vibrant tapestry. 1. Crossing over. This genetic recombination, occurring in prophase I, creates chromatids that are a mosaic of maternal and paternal alleles. It shuffles the genes within chromosomes, creating novel combinations that would not exist otherwise. It's like taking two decks of cards, shuffling them together, and then dealing out new unique hands. 2. Independent assortment of homologous chromosomes. The random orientation of homologous pairs at the metaphase plate in meiosis I means that each gamete receives a completely random mix of maternal and paternal chromosomes. For humans with 23 pairs of chromosomes, there are over 8,223,000 possible combinations of chromosomes in a single gamete, purely from independent assortment alone. This shuffles the genes between chromosomes. 3. Random fertilization. Beyond the internal mechanics of meiosis, the final layer of diversity comes from the sheer chance of which particular sperm fertilizes which particular egg. Given the immense number of unique sperm and egg cells, the probability of any two individuals, other than identical twins, being genetically identical is astronomically small. It is the ultimate lottery of life, a beautiful roll of the genetic dice. Why is this genetic diversity so vital, you might wonder? Why does nature invest such intricate energy in shuffling and reshuffling the genetic deck? The answer lies in the very resilience and adaptability of life itself. In a constantly changing world, environments shift, diseases emerge, and challenges arise. A species with a wide range of genetic variations within its population is far more likely to possess individuals with traits that allow them to survive and thrive under new conditions. Diversity is the raw material for natural selection, the engine of evolution. It is life's insurance policy, its promise of enduring vitality. So you see, while mitosis ensures the faithful continuation of the individual, meiosis ensures the vibrant, ever-evolving continuation of the species. It is a biological masterpiece, a profound dance of reduction, and recombination that sculpts the endless forms most beautiful and wonderful. It is the hidden engine of individuality, the quiet whisper of adaptation, and the very heart of life's enduring, spectacular diversity. Next, we shall delve deeper into the very blueprint of this inheritance, exploring the structure and function of the molecules that carry these intricate genetic instructions, the nucleic acids themselves. But for now, let us simply marvel at the elegance of meiosis, the quiet revolution within the cell that gives rise to all the vibrant, unique life we see around us. We have journeyed through the intricate dances of cell division, marveling at how life perpetuates itself, how new cells arise from old, and how the exquisite ballet of meiosis orchestrates the grand symphony of genetic diversity. We've seen the chromosomes, those macroscopic carriers of inheritance performing their synchronized movements. But now, my curious companions, let us quiet our gaze and delve deeper, beyond the visible choreography of the chromosomes, into the very essence of that inheritance. Let us explore the whispers, the silent instructions, the molecular blueprint that guides every facet of life's intricate design. We speak, of course, of the nucleic acids, DNA and RNA. They are the ancient scribes, the master architects, holding within their delicate structures the complete story of an organism, from the smallest bacterium to the towering redwood, to you and me. Imagine, if you will, an immense sacred library, not of paper and ink, but of shimmering molecular strands, each containing volumes of information, meticulously catalogued and protected. This, in essence, is the realm of DNA. Let's begin our contemplation with Day, the undisputed keeper of the archives. We've touched upon it before, a double helix, a twisted ladder of unparalleled elegance. But what is this ladder made of? What are its rungs, its rails, and how does it manage to store such an astonishing wealth of information? Each rung and rail of this molecular ladder is constructed from repeating units called nucleotides. Picture a nucleotide as a tiny three-part sculpture. First there is a phosphate group, a sturdy negatively charged anchor. Then a 5-carbon sugar, which in DNA's case is deoxyribose, a subtle hint embedded in the very name of the molecule. And finally, the most intriguing part, a nitrogenous base, a flat ring-like structure that carries the actual coded information. There are 4 distinct nitrogenous bases in DNA, each with its own unique molecular personality. Adenine, A, Pum, guanine, G, Tim, cytosine, and Weissas, thymine, T, Tor. Think of them as the 4 letters of life's alphabet. These bases are not arbitrary. They possess an exquisite selectivity, a profound preference for specific partners. Adenine, with its graceful molecular shape, always seeks out thymine. They embrace in a gentle yet firm hydrogen bond. Guanine, in turn, always finds cytosine, forming an even stronger 3-point hydrogen bond. This unwavering loyalty, this precise pairing, A with T, G with C, is known as complementary base pairing, and it is the absolute bedrock of genetic information storage and transfer. Now let's visualize our ladder again. The 2 long rails are formed by alternating sugar and phosphate groups, linked together in a strong phosphodiester backbone. These backbones provide the structural integrity, the unyielding framework of the helix. The nitrogenous bases project inward from these backbones, forming the rungs, always paired AT or GC. And here's another layer of molecular sophistication. These two strands run in opposite directions like lanes on a highway, one going up and the other down. We call this anti-parallel orientation, and it is critical for how DNA is read and replicated. So we have this magnificent double helix, a spiral staircase of information, where the sequence of A's, T's, C's and G's along one strand precisely dictates the sequence on the other. This elegant arrangement ensures that the genetic message is not only stored securely, but also can be perfectly copied. And copied it must be with breathtaking fidelity and speed every single time a cell divides. Remember our discussions of mitosis and meiosis? Before a cell can embark on its journey of division, its entire genetic library, its complete DNA, must be duplicated. This process, my friends, is called DNA replication, and it is one of the most astonishing feats of molecular engineering in the living world. Imagine for a moment that sacred library we spoke of. If we needed to make an exact copy of every single book, how would we do it? DNA employs a strategy of such brilliant simplicity and efficiency that it truly inspires awe. It's called the SARS semi-conservative model of replication. This means that when a new DNA molecule is formed, it consists of one original parental strand and one newly synthesized daughter strand. Each new helix is a hybrid, a testament to its heritage. How does this molecular marvel unfold? Let's gently pull back the curtain on this intricate ballet. The process begins with an enzyme called helicase. Picture it as a molecular zipper, slowly and deliberately unwinding the double helix, breaking those gentle hydrogen bonds between the complementary bases. As it moves, it separates the two strands, much like unzipping a jacket, creating a replication fork, a Y-shaped region where the new strands will be built. Once the strands are separated, they become templates, guides for the construction of new partners. Here enters the star of the show, the enzyme, DNA polymerase. This magnificent molecular machine glides along each exposed template strand, reading the sequence of bases. With unwavering precision, it recruits free-floating nucleotides from the cellular environment, and following the strict rules of complementary base pairing, A always with T, G always with C, it links them together to form a new complementary strand. But DNA polymerase is more than just a builder. It is also a meticulous proofreader. As it adds each new nucleotide, it checks its work, ensuring that no mistakes have been made. If an incorrect base has been inserted, it swiftly removes it and replaces it with the correct one. This self-correction mechanism is vital, minimizing errors and maintaining the integrity of the genetic code. Imagine a scribe who not only writes with incredible speed, but also instantly corrects any typo before moving on. Now, recall that anti-parallel nature of the DNA strands. This creates a fascinating challenge for DNA polymerase. It can only synthesize new DNA in one direction, from what we call the 5' to the 3'. This means that one of the template strands, the leading strand, can be copied continuously in one smooth, uninterrupted flow as the replication fork opens. It's like a train moving forward on a clear track. However, the other template strand, the lagging strand, presents a different scenario. Because it runs in the opposite direction, DNA polymerase must synthesize it in short, discontinuous segments. These tiny fragments, known as Okazaki fragments, are like small bursts of writing. Once each fragment is complete, another enzyme, a DNA ligase, acts as a molecular glue, sealing these fragments together to form a continuous new strand. It's a beautifully coordinated effort, ensuring that both template strands are fully and accurately replicated. The speed and accuracy of DNA replication are truly astounding. In human cells, billions of base pairs are copied in just a few hours, with an error rate of perhaps one mistake in every billion nucleotides. This level of precision is absolutely crucial, for even a single misplaced base can alter the genetic message, potentially leading to significant consequences. It is this faithful copying that ensures the continuity of life, passing the precise instructions from one generation of cells to the next, from parent to offspring. So, DNA is our stable long-term genetic archive, securely housed and meticulously duplicated. But how is this information, this vast library of instructions, actually used? How does a cell read these blueprints to build the proteins that do all the work? This is where DNA's close cousin, RNA, steps onto the stage. While DNA is the master blueprint, RNA is the versatile working copy, the messenger, the translator, the temporary instruction manual. It shares many similarities with DNA, but also possesses distinct features that allow it to perform its diverse roles. Like DNA, RNA is also a polymer of nucleotides. However, there are three key differences. First, the sugar in RNA is ribose, not deoxyribose. Second, instead of thymine, T, RNA contains uracil, U, which still pairs with adenine, A. And third, while DNA is typically a double helix, RNA is usually single-stranded. This single-stranded nature allows RNA molecules to fold into complex, often temporary, three-dimensional shapes, enabling them to perform a variety of catalytic and structural functions. There are several types of RNA, each with a specialized role in expressing the genetic information stored in DNA. We have messenger RNA, mRNA, which carries the genetic code from the DNA in the nucleus to the ribosomes in the cytoplasm, like a delicate scroll bearing a vital message. We have transfer RNA, tRNA, tiny cloverleaf-shaped molecules that act as molecular couriers, bringing the correct amino acids to the ribosome during protein synthesis. And we have ribosomal RNA, rRNA, which forms the core structure of ribosomes themselves, the cellular machinery where proteins are actually built. The journey from the stable archive of DNA to the functional proteins that animate life is a magnificent multi-step process, often referred to as the central dogma of molecular biology. It is a story of transcription, where DNA's code is copied into RNA, and translation, where RNA's message is used to assemble proteins. As we conclude this exploration of DNA's structure and its miraculous replication, let us pause and reflect on the profound elegance of these molecular mechanisms. The double helix, a symbol of life itself, is not merely a pretty shape. It is a testament to perfect design, a structure optimized for information storage and faithful transmission. And DNA replication, with its molecular zippers, builders, and proofreaders, is a silent, continuous miracle, happening billions of times a second across the living world, ensuring that the legacy of life in all its intricate detail continues unbroken. Next, we will unravel how these silent instructions are read, interpreted, and ultimately brought to life through the dynamic processes of gene expression. Beginning with the transcription of DNA into RNA, it is a story of how the blueprint becomes the bustling living reality.